All right, today we are going to start talking about the cell cycle. So this is coming from chapter 12 of Campbell's biology. We'll start right in with concept 12.1. So most cell division results in genetically identical daughter cells. So when cells go through replication and they're going to divide, usually the cells are producing an exact copy of themselves or a clone of themselves. So both daughter cells that results when a cell divides have identical genetic information. There is an exception to this, and that is in the process that we call meiosis. And this is a special type of cell division that is only used to produce sperm and egg cells. And we'll cover that in the next chapter. So all of our DNA is collectively called our genome. That genome can consist of a single DNA molecule, which is what happens very commonly in prokaryotic organisms such as bacteria and archaea, and they often have one singular and oftentimes circular piece of DNA. Or it could be a number of DNA molecules. And this is what happens in eukaryotic cells. Eukaryotic cells typically have several linear pieces of DNA that together constitutes our entire genome. When we take DNA and we package it up prior to dividing the cell, or even when we have the DNA just loosely packed, we would refer to those pieces of DNA molecules as chromosomes. So those chromosomes are a complex thing that consists of both DNA and also the proteins that are responsible for packaging it. Remember that an individual cell, at least one of our cells, has about 2 meters worth of DNA. It needs to be packaged up very tightly in order for us to actually fit that inside of the nucleus, inside of a single cell. So each species has its own characteristic number of chromosomes in the nucleus. And eukaryotic organisms typically, or at least multicellular organisms, are usually divided into two types of cells. So there are somatic cells that are responsible for normal activities. This is our muscle cells, our brain cells, our liver cells, and they typically have two full sets of chromosomes. So one from mom and one from dad. Our gametes, on the other hand, have exactly half the amount of DNA that all the other cells have. And we'll talk more about that later. All right. So here is a picture of a set of sister chromatids. So these are two chromosomes that happen to be identical copies of a singular piece of DNA. So we have one double-stranded piece of DNA here in this linear piece, and then we have an exact copy of it over here, and they are closely bound together. These are closely bound together because the cell is going to go through a process where it wants to split the DNA evenly in half. And this is one copy of this piece of DNA, and here's the other copy. So we need to keep these two together until the point at which we are about to separate. Otherwise, we will end up with a mismatch of which chromosome each cell gets. These are connected together all along the length through proteins called cohesions. But there is also a special point somewhere around the middle, it's oftentimes offset, called the centromere, where these two chromatids are even more tightly connected, and this is where the cell will connect to in order to ultimately pull these two sister chromatids apart. Now, during cell division, the two sister chromatids each are duplicated, and then they separate, and they're going to move to the nuclei of the two daughter cells. So once these chromatids are separated, we would refer to them again as chromosomes. So they're called chromatids when they're linked together, chromosomes when they're their own thing. So as a review of this process, here we have a cell. It has this piece of DNA in it. The very first thing that's going to happen is that DNA is going to be replicated. Once that DNA is replicated, we now have two copies. Those are going to be bundled into these things called chromatids, and they're going to be linked together. Ultimately, when the cell divides in two, each one will have a copy of that piece of DNA. And recognize that we have several pieces of DNA, and this same process is going to happen for each and every single piece of DNA that we have. So again, concept 12.1 is most cell division results in genetically identical daughter cells. So we have the mother cell starting off with a singular piece of DNA. Both daughter cells are going to look exactly the same as the original mother cell.